TRP operon is a set of genes in bacteria such as E. coli that control the synthesis of the amino acid tryptophan. TRP operon contains 5 structural genes which are TRPE, TRPD, TRPC, TRPB, and TRPA which encode tryptophan synthase. It also contains a promoter where RNA polymerase binds to a repressor gene which synthesizes a specific protein. Then it will bind to operator to block transcription. Tryptophan is only synthesized when it is not available in the environment, allowing the TRP operon to preserve energy. As a result, it is a component of the anabolic pathway which produces molecule. On the other hand, the catabolic pathway is regulated by the lag operon which release energy for biological activity by breaking down complex molecule. Next, Wafi will be explained further about repression.
Thank you, Dalina. Now let's discuss how repression works in the TRP operon, which you can see illustrated in the diagram. In the absence of tryptophan, the regulatory gene TRPR, which encode inactive repressor, so it can't bind to the operator. Because of that, RNA polymerase can bind to the promoter region, initiating transcription of the structural genes. These genes produce enzymes needed for tryptophan synthesis. In this state, this operon is derepressed, meaning these structural genes are expressed. However, when tryptophan levels in the cell are high, tryptophan acts as a co-pressor by binding to the repressor protein. This binding activates the repressor, causing it to change shape and enabling it to bind to the operator regions on the DNA. When the active repressor binds to the operator, it blocks RNA polymerase from transcribing the structural genes. So that means this operon is repressed. As a result, the enzyme required for tryptophan synthesis are not produced, preventing unnecessary energy and resource expenditure. This negative feedback mechanism ensures that tryptophan is synthesized only when needed. To elaborate further, the TRP operon system demonstrates how bacteria efficiently regulate their metabolic needs. This repression mechanism ensures that resources are not wasted on producing tryptophan when it is already present in sufficient quantities. The switch between the inactive and active states of the repressor protein highlights a sophisticated level of control in prokaryotic genes expression. For instance, if the environment suddenly lacks tryptophan, the repressor protein quickly becomes inactive, allowing RNA polymerase to resume transcription. This adaptability showcases the operon's dynamic nature and its crucial role in bacterial survival and efficiency.
Okay, for the next part, we are going to discuss about the second regulatory mechanism, which is attenuation. Attenuation system decreases the gene expression, same as repression does, but attenuation does the process while the RNA polymerase transcription is still in progress. So there is a gene sequence which is called TRPL that present in the beginning of the transcribed gene, which has at least 130 nucleotides that will include the 1,4 sequence, which partially correlate to each other. So when the condition where tryptophan is abundant, the tryptophan charged tRNA happens to be abundant too, making it to be always available for the ribosome to continue translating the mRNA until it reaches the region 2 of the TRPL. So this condition allows the formation of the 3-pair hairpin to form, making the termination process to happen. So when tryptophan is in scarce, ribosome will pause its translation at sequence 1 because of the lack of tryptophan charged tRNA, and this will result in the formation of 2-3 hairpin to form. So 2-3 hairpin formation prevents the 3-4 hairpin to form, which this condition allows the RNA polymerase to continue the transcription process.
Thank you for the explanation, Faris. Next, we're moving on to the combination of repression and attenuation. By combining these two processes, they can regulate the transcription of the tryptophan operon in response to different levels of tryptophan in the cell. This is because repression happens at the initial step of transcription in response to high tryptophan level, whereas attenuation regulates the termination of transcription in response to low level of tryptophan. This combined mechanism ensures that expression of TRP operon is turned off when tryptophan levels are high. will reduce the transcription initiation, whereas the attenuation will adjust the level of transcription that escape repression, which are acting as fine-tuning mechanism. For example, when the levels of tryptophan is high, the repression mechanism reduces TRP operon transcription, whereas attenuation can add another reduction to the remaining transcription, ensuring the synthesis is tightly regulated to meet cellular need without overproducing amino acid. The mechanism is efficient as the mechanism inhibits inefficient transcription and promotes maximal expression depends on the level of tryptophan. Comparison of mechanism between TRP operon and ARA operon. The regulation in TRP operon focus on synthesizing tryptophan efficiently and can avoid waste through attenuation, while the ARA operons prioritize breakdown of their respective substrate only when needed. Next, for the role of regulator. The TRP operon have repressor that inhibit transcription when bound, whereas for ARA operon, the regulator can act as repressor and inhibitor. Last but certainly not least is the additional regulation. For TRP operon, they have attenuation, whereas for ARA operon, it requires for CRP-CAMP for full activation.
Thank you, Aisyah. So let me tell you a little bit more about trioperon. Trioperon gives significant importance and have its own application. The first one, trioperon conserve energy of bacteria. Learning about trioperon gives us an understanding about how bacteria adapt to the surrounding. By regulating the trioperon's expression according to the tryptophan levels, bacteria can more effectively distribute their metabolic resources. When tryptophan is in short supply, the operon is activated, enabling the bacteria to synthesize it. When it is abundant, the operon is repressed, saving energy that would otherwise be used for biosynthesis. As we know, trioperon is a system that produces amino acid called tryptophan. Tryptophan gives many benefits to the human, especially in pharmaceutical sector. Tryptophan used as a supplement to treat multiple illness because it works as a precursor for melatonin and serotonin. For melatonin, it can help in regulating sleep-wake cycle, while serotonin is beneficial for those who have anxiety, depression, or mental disorder. Tryptophan has also been used in the agricultural sector, since it also can act as a precursor for auxin hormone that very important for plant. Auxin promotes cellular differentiation of vascular tissue, especially in shoots and mediates vascular tissue regeneration after wounding and makes healthy crop of plants. Thank you. Thank you, Aisyah. So let me tell you a little bit more about trioperon. Trioperon gives significant importance and have its own application. The first one, trioperon conserve energy of bacteria. Learning about trioperon gives us an understanding about how bacteria